So thanks again for the introduction. Today I want to talk a little bit about variants in thyroid hormone transporter MCT8 and how they relate to disease outcomes and some other stuff. And indeed, I have my PhD in the Erasmus MC in Rotterdam over here, and I'm currently working as a resident in internal medicine in Delft, and that's where I'm actually right now. So I have to disclose that I do fulfill a consultancy role for Geest Therapeutics, which is the marketeer of a drug called Triac. So to start off with the basics, the thyroid is a gland within the neck which produces thyroid hormone, and thyroid hormone is important for energy balance and for brain development early in life. And the thyroid gland is regulated by the pituitary, which is up in the brain with a negative feedback loop, as we often see in endocrinology. And thyroid hormone has to enter the cell by active transport through thyroid hormone transporters. And when it is inside the cell, it gets converted and eventually, in the end, it binds to its receptors and there has its genomic actions. So it's a complex system that has to go according to the plan. And a very important thyroid hormone transporter is the monocarboxylate 8 transporter, or MCT8, which is particularly relevant in the brain of human individuals. So it allows thyroid hormone to enter the brain of individuals. And when there's mutations in MCT8, we see a very particular phenotype, a very rare disease, I have to say. So there's approximately 500 patients across the globe, and we see increased serum T3 levels. So T3 is the biological active thyroid hormone, decreased T4 levels, and normal TSH levels. But most prominently, we see very severe intellectual and motor disability and signs of peripheral thyrotoxicosis, meaning that there's too much thyroid hormone. And that makes sense to us because MCT8 is very important in the brain. The high thyroid hormone levels can't enter the brain and thereby result in severe intellectual and motor disability. On the other hand, we know that organs like the heart and the liver, those peripheral organs, do not require MCT8 to transport thyroid hormone into the cell. And as I mentioned, the serum levels are elevated, so these tissues are in a chronic peripheral thyrotoxic state. This disease is X-linked, so males are affected predominantly. In our group, we evaluated the phenotype of this very rare disease, and we did that by combining clinical data from patients from many places of the planet, basically. And what we then see is that they all have very low GMFM scores, which are a measure for development of the brain. Many of them are not able to hold their head up independently, so it's a very severe phenotype. Often they have spasticity, and we also see that many of them are underweight. So again, very serious disease. And we also see this coming back in mortality rates. You can see here, and I hope you all appreciate that the Kaplan-Meier curve is going down very rapidly. You can see that patients die early in life. Many of them don't make it to 10 years of age, unfortunately. When we do dive a little bit deeper into this data, we can classify in two groups, and we see that those patients who are able to control their own head, basically, so who are able to hold up their own head, have way better survival than those patients who do not have head control. So there's phenotypic differences. Similarly, we see that those patients that are underweight early in life, here in blue, die much younger than those patients who are not underweight. So knowing this, we wondered, why is there this difference in this clinical presentation, with some patients even being able to walk some steps by themselves, but most patients not having any head control, and similarly with most patients being severely underweight and some patients having a normal weight. And we wondered, is there a genotype that is underlying these phenotypic differences? So in the project that we did, we aimed to unravel those genotype-phenotype correlations. And in order to do that, we used our international consortium and did a full review of the complete literature. So we ended up with close to 400 patients, which we believe is the biggest number of these patients that we can find. And we did several functional studies in our lab using thyroid hormone transport assays with radioactivity in overexpression models, but also in patient-derived fibroblasts. And we combined these data to search for genotype-phenotype correlationships. So we tested 130 unique variants within the MCT8 protein all throughout the different exons. And then we separated the data in 3 different groups. So we made a group of mildly affected residual uptake capacity, moderately affected residual uptake capacity, and severely affecting mutations. And what we then see is that there's very clear correlations with the uptake capacity and phenotypic outcomes. So here again, I show survival plots, and you see that those patients who have a severe mutation die much earlier in life compared to those with a moderate mutation or with a mild mutation. And similarly, we see this coming back in, again, these GMFM-88 scores with severe patients hardly having any development and mild patients showing some development. And it's good to realize that 100% GMFM score basically converts to a healthy 4-year-old child. So still, these patients are quite severely affected. Again, we see something similar for outcomes like head control and sitting and walking here in red. I think you all can appreciate that most patients do not have any head control at all, whereas those with a mild mutation do have much more head control. We see something similar for seizures, and actually we find these correlations with over 25 phenotypic outcomes, not only on mortality and neurological outcomes, but also on things like body weight and heart rate, etc. We do not only see this in overexpression models, but also in patient-derived fibroblasts. We had a smaller number of them, but clearly you can see here that those with severely affecting mutations die earlier than those with mildly affecting mutations. And again, the GMFM scores show this very clearly, I think so. So after this, we were wondering, might there also be some sort of mild resemblance of this very severe phenotype on a population level? So we collaborated with people from the Thyroidomics Consortium and from the UK Biobank in order to investigate whether single nucleotide variants in MCT8 show any significance with such phenotypic outcomes in healthy individuals. And indeed, to go somewhat shortly, we find that there's a resemblance of this phenotype on a population level. And I here highlight 2 particular SNPs. You can see that free T4 is correlated, a lower free T4 with these SNPs. And also we find brain outcomes on MRI scans of particular regions that we also see in our actual patients that are smaller. We also find that in these GWAS data. Subsequently, we were wondering whether we could also evaluate the effectiveness of a therapy that we have developed. And what you basically want to do in this disease, so there's 2 problems. On the one hand, you want to increase thyroid hormone action in the brain because there's a lack of thyroid hormone in the brain. But on the other hand, you have to decrease thyroid hormone action in the peripheral tissues because there's too much thyroid hormone going on. And TRIAC, which is a thyroid hormone analog, seems to be able to do this. And since it's a TRIAC hormone analog, it can bypass MCT8 but still enter the cell via other transporters. But once it is inside the cell, it can bind to the receptor of thyroid hormone and act in a very similar way. And it's also degraded in a similar way to T3. So ideally, this is a very suitable drug. And indeed, we found in clinical studies that if we treat our patients with TRIAC that we can alleviate this peripheral thyrotoxic phenotype. So here in blue, you see the baseline serum T3 levels being clearly elevated in the majority or basically all of the patients. And when we treat them for 12 months, you can see that serum T3 normalizes. Moreover, we see that important clinical outcomes like body weight improve upon treatment. And we've done additional studies to evaluate the long-term effects of TRIAC, and we see very similar findings. And currently, we are evaluating whether TRIAC treatment in very young individuals can also save the brain development, yes or no. And based on these data, the European Medicine Agency recently approved TRIAC as a drug for treatment of the peripheral thyrotoxicosis of MCT8. We were then wondering, is there a difference in those patients with a severe mutation versus those patients with a mild mutation when it comes to treatment? And basically what we see is that the reaction of patients being treated with severe mutation versus with a mild or a moderate mutation are very similar. So you see that the serum T3 levels are going down. And not only serum T3 levels, but also the positive outcomes on body weight, heart rate, and other endocrine markers show exactly the same pattern across severe and moderate and mild patients. And to us, that makes sense because we believe that TRIAC is transported not via MCT8, so we expect very similar results. So after doing this, we were wondering, can we better understand what exactly is going on in the protein? And can we identify critical regions within MCT8 so that we also understand what novel variants might do in patients and whether they are disease causing or not? So we performed an alanine scanning of this entire protein. So basically replacing every amino acid with an alanine and performing functional testing again with T3 and T4 assays. Here I show the conservation of MCT8. So the N-terminal tail is quite conserved, but doesn't seem to have an important role in the protein's function. But you see that mostly the transmembrane domains are highly conserved. And when we then plot our alanine results to this, with here in red and orange, you see the most severe impact of an alanine substitution. I think you can see a pattern that particularly the transmembrane domains seems to be important for the function of MCT8 in thyroid hormone transport. And we all plotted this on an homology model. And I think here you can see, so here in light blue, I hope you can see it, is actually the thyroid hormone plotted in the graph. And you can see that particularly on the inside of the protein, there's much red and much yellow, indicating that are the regions that are very important for thyroid hormone to be transported, whereas the outside of the protein is much less relevant, which makes sense. By combining this, we were able to identify 7 critical regions within the protein, of which many were not identified before. Many are across the substrate binding pocket, but also there's 2 regions at the bottom of the protein, basically, that serve as a linker region between the 2 halves of the protein. So this gives us new insight on how the protein works, and also what this might mean for future variants. I want to highlight one particular amino acid residue, which is the N193. So here in this graph, you see that the vast majority of variants have a very similar impact on T3 and T4 impact, but this one in red, which of course is the N193, has much more impact on T4 uptake than on T3 uptake. And when we zoomed in on this particular residue, we found that thyroid hormone really comes close by within the transport process. We did some additional analyses, and then we find that not only T4 has more difficulty to be transported, but also reverse T3. And when we then look at the structural properties of these hormones, we see that those hormones, being T4 and reverse T3, that have this particular iodine moiety over here, cannot be transported, whereas those molecules that do not have this iodine moiety and otherwise very much are alike T4 and reverse T3 are able to be transported by this particular variant. And very recently, in collaboration with a group in Milan, we were able to create a crystal structure of MCT8 based on also based on the findings that I previously showed. And now we much better understand the actual transport process of MCT8 and also of very well-known mutations. And also now we better understand how the particular inhibitor of MCT8 works. After this, there was one thing that we wanted to do as well, and that's also, well, I think important for today. So we want to see if we could classify pathogenicity and severity of novel variants in MCT8. And we did this by collaborating with Jonathan Fraser and Mafalda Diaz, who have designed EVE. So we used unsupervised AI approaches and using sequences of the tree of life. And we can already see that the EVE can quite clearly distinct between mildly and severely affecting mutations when we compare it to our uptake data. Then we took it a step further and we also started to include structural and chemical properties. And we did this together with Mariano Martin, who's also here today, and build basically a supervised dual classifier in which we aimed to classify pathogenic versus non-pathogenic variants. And with our MCT8 classifier here in red, we were able to quite well distinguish pathogenic from benign variant. And actually, we're doing that much better than commonly used tools like SIFT or Polyphen, which are currently used in clinics when new variants are observed in patients who are suspect of MCT8 deficiency. And also, our combined MCT8 deficiency classifier outperformed the EVE tool by itself. Subsequently, we wanted to evaluate whether we could also distinguish between mild and moderately and severely affecting pathogenic variants. And you can see here, I think, that we were able to do this somewhat reasonable and also that we're outperforming EVE here. The other classifiers like Polyphen and SIFT do not provide a severity classifier, so we can't compare it to that. And in the end, we were able to basically build a mutational landscape heat map. I didn't put in all colors in the entire protein, but we have it for basically every residue in which we predict whether an amino acid change at that particular position is pathogenic, yes or no, also dependent on what amino acid is inserted. We did some small experiments in our lab to validate whether this classifier works or not. And indeed, those variants that were predicted to be pathogenic all had a T4 uptake of less than 50%, which we would consider pathogenic. And those that were predicted to be benign, the majority of them indeed were benign, with exemption of two variants. When we compare this again to Polyphen and SIFT, we see lower accuracy in Polyphen and SIFT. You can see here that this entire row is predicted as damaging, but there's 3 variants that are actually benign. And you see something very similar here in SIFT. So our pathogenicity classifier outperforms Polyphen and SIFT. When we do some similar evaluations for our severity classifier, you can see that we're having somewhat more difficulty, but we are somewhat able to distinguish mild from moderate and severe loss of function mutations. So in conclusion, we have now learned that the functional impact of MCT8 mutations correlates with severity of more than 25 disease features, and we find a mild resemblance of this phenotype on a population level. We now know that the triac therapy is independent of mutational status in its effectiveness, and we've identified 7 critical domains within the protein, and we've built a pathogenicity classifier which can really help us in clinic in the future, and our severity classifier somewhat distinguishes between mild and moderate and severe loss of function variants. And with that being said, I would like to thank everyone that's been involved in this, and I'm happy to discuss further.
Okay, great. Ferdy, great talk. If you have any question from the audience, you can write it down in the QA or in the chat. And well, we don't have any, but I have some. So triac is the mechanism of action of triac is independent of MCT8, right?
Yeah.
So it works independently of the mutation that the patient have.
Yeah. So we believe that triac is transported with other transporters than MCT8, thereby making it independent. Yeah.
Okay. And have you considered to use some small molecule like a pharmacological chaperone or something like that that may stabilize MCT8 mutants, so this improves the T4 and T3 uptake?
Yeah, excellent question. That's something that is being done in the field as well. So it depends on, and I didn't go into this right now, but it depends on the mutation, whether the mutation is indeed destabilizing the protein or if it's something else. But for particular variants, it is a possibility, and it has also been done in some patients with these variants, with not very clear effects, although the numbers have been very small. I think approximately 4 or 5 patients have been treated. We do know that the chaperone therapies that are commonly applied can be quite a problem for the liver, and the liver is already affected in these patients. So you have to be careful with these treatments, but it is something that is being evaluated in the field as well. Yeah.